Just PR. They haven't decided what level it's at.
Dr. Na Chai was an undergraduate here on the Downing site a remarkably short time ago. You see, she's very young. She did a PhD then in Oxford and then a postdoc at the Sanger Institute. And she's now already a PI in Munich.
Thank you. Thank you so much. Good afternoon, everyone. Thanks for coming. As Professor Mollen just said, I'm a group leader at Helmholtz now, and ever since I left Cambridge, I've always tried to come back, and we stayed in touch, and I'm very grateful that he gave me this opportunity to come to talk to all of you. I work on genetics of depression, and that's what I'm going to talk about today. So a lot of it is going to be methods that I use to investigate genetics of depression, and also some of it is lessons that we've learned from all the analysis that we've done so far. So I hope you find it interesting, and I hope I don't go too much over time, because there's a lot to cover. So why study depression? Well, maybe for this audience, this is preaching to the choir. You all, I guess, study psychiatric diseases to some level. But the reason why we want to study depression is it is a very common and debilitating disorder that currently cannot be well diagnosed or treated. There's no measurable biomarker for diagnosis. We don't know the biological pathways leading to it, and treatments are pretty much trial and error. They mostly fail in the first instance. So needless to say, there is an urgent need to make the situation better. And ideally, that would be through understanding the biological pathways leading to depression, and then using that, we can identify biomarkers for diagnosis as well as drug targets that can be more targeted in treating the disease. And that's really the ultimate goal of genetic studies of depression. The more more interim goals that are on this slide may be achieved in more achievable in individual studies. And that is to identify genes that contribute to depression etiology, the biological pathways that they're part of, maybe the tissues in which they act, and also whether they can be good drug targets if we get that far. We also need to figure out if these genes, pathways, or tissues correspond to certain clinical characteristics of depression that we can observe in people as they come into the clinic, so that we may be able to triage them, we may be able to diagnose them, and point them to specific types of treatments that may be more effective for them. So that's some of our slightly shorter term goals, other than to just understand nature of depression. So how it all began? Well, it began sometimes in the 70s and 80s. So genetic studies on depression began around then. And these studies have taught us the first lessons on depression genetics. So first came family studies, which basically asked the question, what is the chance a person will get depression if they had a family member who has depression, versus someone without family members who have depression? And some of these early family studies showed that the odds of getting depression, if you have a family member who does, is around 2 to 4-fold of someone who doesn't. And this told us that depression, to some degree, runs in families and is at least likely to be partly induced by genetics. Then we had twin studies, which looked into the difference in the disease correlation between monozygotic and dizygotic twins. And using the genetic sharing between monozygotic twins and dizygotic twins and the difference between them, they were able to work out the proportion of disease liability that is driven by genetic factors. And so all of these early twin studies have sort of converged on a number of around 40%. So 40% of depression liability has got to do with genetics, putting it quite close to some of the other common disorders that we know about, diabetes, etc. And then what happened when small-scale genotyping or sequencing became possible, there is a period of time, a dark history of genetics, where Canada gene studies took off. So here what happened was people were collecting relatively small groups of people through their clinics, and they were genotyping them based on hypotheses on the biology behind depression. So they were asking if patients, these patients that they've collected, they more often have a particular genetic variant than those without the disease. So this is a review published quite recently, 2018, reviewing all of the candidate gene studies that has been done. And each of these bars is the number of studies that has been done. On the x-axis is all the genes. I think I can point to. It's all the genes that have been studied. The most popular one here is a serotonin reuptake, like serotonin receptor. And the height of this bar is the number of studies that has been done on it. And the orange part of it is number of studies done on the most popular variant, not the gene, but the actual genetic variant. So you can see that a lot of studies have gone into this, a lot of resources and man hours. But unfortunately, as most people in this room might know, this approach is wildly unsuccessful. These genes and variants shown in the previous plot have now been tested for association in much larger populations, much larger data sets where we have genetic data on all of them. And here's on the y-axis is the odds ratio, on the x-axis is the genes and the variants that have been tested in candidate gene studies, and all of them are not significantly different from one, which means that they have no effect on depression. So first lessons from all of these early studies. Depression is polygenic. It's partly genetically driven. Around 40% of its liability could come from genetic factors. But none of this hypothesis-driven research or testing has worked. And the fact that the failure of this sort of persistent efforts in candidate gene studies have also sort of like on the side in one way could tell us that there's very unlikely any common variants of large effects contributing to depression that could be robustly identified with such an approach. So, a bit of a false start for genetics. Try again. And that's when genome-wide association studies came into the picture, especially with the lowering of costs of genotyping and sequencing in the later 2000s. So in a GWAS study, a large group of cases of patients with the disease and controls, usually matched for ancestry, age, sex, etc., are genotyped at hundreds of thousands or sequenced across their genome. And each of these variants that has been genotyped or sequenced is then tested for association with the disease, usually with this model here. It could be linear regression, could be logistic regression, depending on what you use. So on the left-hand side of this equation is your disease of interest, ones and zeros, ones for people who have it, zeros for the people who don't. There's a bunch of covariates you might want to control for, sex, age, etc. And then you have your variant that you want to test. And then the beta is the effect of the variant on the disease itself. And the beta is what we're interested in. We're basically testing for beta not equals to 0 here. And then in doing that, you also need to control for genetic relatedness between individuals. And of course, there's a noise term. So then you do this hundreds of thousands of times or millions of times, depending on how many variants you have. And because you're doing so many tests, the multiple testing burden for statistical significance is pretty high. So we usually use 5 times 10 to the power of minus 8 for the p-value threshold. So then what we're supposed to do for GWAS is then we're getting a bunch of significant variants passing the p-value threshold. And from those significant loci, we then generate biological hypotheses to follow up on. So at the beginning, when GWAS first started happening, again, it's people collecting relatively small cohorts, usually in around 100 or thousands of people. And then they're usually collected through clinicians' interviews, and they're done by individual groups around the world. So GWAS on any one of those cohorts hasn't given any significant findings because of the really high statistical testing burden. So what happened was there's this group of people coming together to form the Psychiatric Genomics Consortium around 2007. And this group is a sort of international consortium putting together all the data collected in various groups around the world together in a meta-analysis. And this was the first meta-analysis published in 2013. It has data from 9 different groups for discovery and a bunch of replication cohorts. The discovery data set has 9,000 cases of depression and 9,000 matched controls. And this plot here is how we normally represent a GWAS. So on the x-axis, you have the genomic position from chromosome 1 to chromosome 22. And on the y-axis, you have the negative log 10 p-value. So the higher it is, the lower the actual p-value, the more significant. And this line here is what I was talking about. This is the significance threshold, and it's quite high. So as you can see from this plot, and each of these dots is a genetic variant that you've tested. So dots above this red line are significant, dots below it are not. And as you can see from this plot, none of the variants are significant. So what this means is that the effect sizes for at all of these genetic variants are too small to be significant at this sample size. They would not give you a p-value that is small at this sample size. So then, what can we learn from this fact? So either the effect sizes are smaller than they should be, because maybe some of these effect sizes are really quite big, but then for some reason, maybe because of noise in the data, etc., they're smaller than they should be, and we can't detect them. Maybe if the effect sizes are all what they are supposed to be, then our sample sizes are just too small. So you either increase the effect sizes by cleaning off whatever noise is available in this, whatever noise is present in this data set, or you just increase sample sizes. Two approaches that could potentially solve this problem. And then, because those are the two known solutions, people have answered them. So the first sort of strategy to solve this issue is to collect a homogeneous cohort, eliminating noise as much as possible. And that's the study that I was involved in my PhD. And it's called the CONVERGE study, which collected only severe recurrent depression cases and also controls who have never had any mental health issues. And all of the participants are of age 40 to 60. They're all Han Chinese ancestry, and they're all clinician interviewed, etc., etc. So this is the most stringently phenotyped data set to date. And we think that what it does is that it really did reduce noise in our data collection process, leading to an increase in power for associations. We've collected 6,000 cases and 6,000 controls, smaller than the meta-analysis that you've seen just now, but we found the first 2 ever genetic associations of depression. And this can be replicated in an independent cohort in China. While we're doing this, other groups have taken a different approach. And their approach is to increase sample sizes drastically. So in 2016, this is the first paper published of its kind. It's by the company 23andMe. Has anyone done it? It's a consumer genomics company. You spit in a tube, they give you your genotypes and a bunch of things that you might be interested in. So in doing 23andMe, you're asked to answer a questionnaire. Do you consent to being part of research? And also part of this questionnaire is, have you ever been diagnosed or treated for depression? They have various versions of it, but basically it's that question. So then they took people who said yes to this question as cases, people who said no as controls, they did a GWAS on it, and they found 17 significant loci with 75,000 cases and 230,000 controls. So just note here that the sample sizes have shot up quite a lot, really drastically from the thousands that we were talking about just now. And ever since 23andMe did this has really opened up new grounds. Everyone's doing this now. So basically, since then, 2018, 2019, people have been pulling together self-report data on whether you had a depression before, you had a diagnosis before, have you gone to the doctor before. Also, they have been pulling electronic health record data, so ICD-10 codes. So these are diagnostic codes that doctors give you when you have been diagnosed with depression. And depending on the health system, this could slightly differ between populations. So with the different types of data that is coming in, we've seen a massive increase in sample sizes. This study in 2019 has 264,000 cases, 500,000 odd controls. I know that the latest, actually, in the next slide I have another one. So this one is in 2021. I think it's more than 1 million now. So this is from the Million Veterans Project in the US. They've pulled together depression defined by electronic health records and ICD-10 codes, together with depression by surveys, etc. They've done 2 GWASs on that. And all the sample sizes are above 1 million now. And they've gotten around 180 significant loci. And what I know now of the ongoing situation in terms of ongoing analysis in the community is that the sample sizes have gone beyond 2 million, and the number of hits is double that, so around 300 hits. So we've got loads of hits, right? Now, these are statistical associations with genetic variants. some effort needs to be made to link them with genes, pathways, tissues, etc. Now, these are just variants. It's a totally other job to link it up with other things, to biological things. So the first problem is identifying the genetic variant that actually matters. So we have, like all of these hits that I've talked about just now, they are associations on what we call a genetic locus. So what happens is there's actually a lot of variants in this locus that are associated with your disease of interest. And here, represented pictorially, the two red ones are actually the ones with real biological effect on the disease. But they're not the ones with the lowest p-value. That one is. So then, there's some effort that needs to be made to really identify these two from these ones, which for some reason has a lower p-value because of their correlation structure with these two that have real effects. So this process is called fine mapping, and there are statistical methods to do this. There are various methods to do this now, but they often narrow down the locus down to a few variants which are called confidence SNPs of being causal. They have a high probability of being causal. So this is one of the first steps that you need to take if you want to really understand biological function behind some of these statistical associations. The second problem is to link these associations to genes. Even if you have those causal variants, you then need to start linking them to genes. Now, a lot of GWAS hits are not in genes themselves. They are in regulatory regions, they may be in non-genic regions, they may be somewhere else. And even if they were in genes, if they don't obviously disrupt gene function, it's really hard to tell which genes they're actually working on. So one of the approaches to do this to link genetic variants with genes is through what we call the transcriptome-wide association study, or TWAS. So in TWAS, what happens is the variants that are identified to be associated with the disease are used to predict gene expression from various tissues, given we know the regulatory effects on the gene expression of those tissues. So these predicted gene expressions are then tested for associations with the disease you're interested in, and then that gives us a list of genes that are associated with the disease rather than the variants. So that's another way we could identify genes from all of these studies. So here I show what you can get from applying both fine mapping and TWAS to the latest GWAS on depression that I've shown you a few slides ago. On the bottommost plot, this is the Manhattan plot that we've just talked about. This is the genetic findings. Each of these dots is a genetic variant, and we have their negative log 10 p-value association for the disease. In this middle plot here, we have only the variants which were defined by fine mapping as confident variants. And on the top ones, these are the genes that have been identified via TWAS to be significantly associated with the disease. And the black lines show situations where a genetic variant is found to be potentially causal, and it can be linked to a particular gene. So then these black lines are really, the probably most confident findings you get from this GWAS. And if we wanted to know further which genetic variants from depression, which tissues these genetic variants are acting in, we can further perform tissue enrichment analysis, leveraging gene expression data that we know from different tissues from other reference data sets. And again, there are many, many methods to do this, and new ones are coming out almost every year now. And so this is another figure from the latest paper on GWAS and depression, where they've done fine mapping and TWAS, and in this figure they've shown the genes with the most significant association and enriched in each of these cell types that they have looked at. So this analysis links genes identified in GWAS with their most likely tissue of action. So now we have both genetic variants, genes, and tissue of action. And that's all great, right? Seems we are getting really close to revealing the biology of depression. And if you carried on doing this with 5 million samples, 10 million samples, et cetera, we include the entire world, then it seems that we'll eventually get all the genes and all the tissues, and we can start assembling biological pathways, and et cetera. How am I doing with time? Oh, but I mean, I've got 40 minutes left in my talk, so you know that there's a plot twist coming. Yeah, it's not quite right. So while all this GWAS is happening, there's another separate effort assessing whether using data from these self-report data, electronic health record, is actually valid. This study, for example, asked the question, what is the relationship between the genetic risk factors for broadly defined depression, what we clinically define as depression using all the stringent clinical diagnostic criteria, depressive symptoms, and this personality trait, neuroticism. And what it found is quite unsettling. It found that 65% of the genetic variance for lifetime MDD, which is the clinical version of depression, was unique to this and not shared by all of these other things. So what that means is that genetic studies that have been performed on these things are not going to get you 65% of the genetic variants that are relevant to these. And that's a big problem. And that also shows us that if, for example, there's a lot of unique genetic variants for this, there's a lot of genetic variants for these, what are we actually looking at from all the GWASs that we've done up to now? So I also decided to look into this, and what I did was using a large data set called the UK Biobank. So I decided to define deep phenotyping and shallow phenotyping in UK Biobank, and let's say the deepest phenotyping that you can do for assessing depression is you perform a structured interview with the DSM criteria, the clinical criteria for diagnosing depression. And the shallowest is self-report diagnosis or treatment measures. So then let's map these phenotypes to what's available in the UK Biobank dataset, which is a 500,000 people dataset collected by the UK. And here we have a self-administered CD short form, which has all of the questions necessary for diagnosing depression using clinical criteria. Now, it's not quite the same because it's not administered by a clinician, but it's the closest we have in UK Biobank. So let's call this the deepest possible in UK Biobank. And it's we've called it lifetime MDD, and it's in purple, and this color will stay for the rest of the presentation. Yeah? On the other end of the spectrum, we have the help-seeking measure of depression. This is, have you gone to the doctor? Have you gone to the GP for depression? And I've called it GP-Psy, and it's in red, and that color stays also. So let's do GWAS on both of these things. So lifetime MDD, there's only 67 people who answered enough questions to be defined this way. You do the GWAS on it, you find one hit. Okay? And then GP-Psy, as with a lot of other shallow phenotyping measures, you can get it like really quickly in a lot of people. It's just answering one question. People are more inclined to do it. So we've got 5 times more samples and 18 GWAS hits rather than just one. Okay. So we've done a lot of analysis trying to figure out whether the two are the same things. They're supposed to be the same thing if they're really all tagging depression. But here, what we've done is we've asked what is the amount of genetics that is contributing to each of these phenotypes. On the far left of this figure, we have lifetime MDD here, and the sort of amount of genetic contribution to lifetime MDD is around 20-something%, 23, 24%, which is lower than the 40% estimated from twins, but is quite high. Here is GP-Psy, and you can see it's significantly lower. It's around 12%. And that's one of the first things that we find. What would cause this to have a much lower genetic contribution than this? So some people are saying, okay, well, it could be a milder form of the disease. But the thing is, this measure here shouldn't change, because if you just took the milder form of the disease. This measure here is how much genetic contributes to liability of the disease. If you just took a milder form, you're really cutting the population at a different place than you would if you took the severe end. The amount of genetics that went into it stays the same. So this number should stay the same. So the milder form argument is just doesn't explain this. We can also look at genetic correlation between lifetime MDD and GP-Psy. So all of these genetic correlation are with respect to this deep measure here. And you can see that GP-Psy has a genetic correlation of around 0.8. So a lot of people say 0.8 is quite high. Like, if something is correlated 0.8, that's pretty good. But these are R values, not R squares. So if you square it, it's 0.64. And that means that what that actually means, in terms of biological interpretation, is that 64% of the genetics effects for lifetime MDD is shared by GP psi. And for two phenotypes that are supposed to be exactly the same thing, 64% sharing is really not a lot. Yeah. So then we can look at GWAS hits for GP psi. There were 18 of them that we saw just now. So here are the effect sizes at all of these SNPs. Okay? So if you look at their effect sizes in lifetime MDD, the first thing you can see that, well, they look quite similar in terms of the shape, right? The effect sizes, some of them are not significant, but at least the effect sizes are in the same directions. Yeah? So that's not bad. But then you can also look at neuroticism, which is not depression. This is a personality dimension. And you can see that the effect sizes are also very similar. The shape is almost identical to that of GP psi. And you can then look at this other phenotype, which is all the people who said they went to the doctor for depression, but they indicated they didn't have any depression symptoms. So what we call GP no dep. And then you can see that the shape is also very similar. So then what does this tell us? This tells us that the effect sizes found, the genetic effects found for GP psi are not specific to depression. They seem to be the same for a huge number of things. Maybe not just the ones that we've displayed here. And then finally, if we did tissue enrichment analysis for these genetic effects, well, one of the arguments for the validity of these kind of self-report measures of depression is that, look, we have CNS enrichment. We have all the brain tissues enriched for effects on these. But I mean, they are also enriched for bipolar disorder. They're also enriched for schizophrenia. They're also enriched for a bunch of other things. So just having tissue enrichments for brain tissues is not a good proof that these are biologically the same thing as clinically defined depression. So what we learned from this range of analysis that we did was that the loci we got so far from all the massive GWASs on depression may not all be specific to MDD. They may be also shared with a bunch of other things. So now that sounds like candidate gene studies again. We don't know anything. This is really bad. Well, the thing is that they're not completely bad, because although I've shown you how the genetic effects are not specific to depression, there is something that these shallow phenotypes do really well in, and that is predicting depression out of sample, surprisingly. So what do I mean by prediction? How is prediction performed? So you have your starting GWAS, which is what we've seen just now, and you take some threshold, some p-value threshold, and you take all of the SNPs, the independent ones that have gone beyond that threshold, and these are the significant ones. You then take the effect sizes at each of these SNPs and multiply that with genotypes in a different cohort and arrive at a score, and that's called a polygenic risk score. It's built from genetic effects found for this disease in a different cohort. You take a test cohort, you get the score, and then what you can do is you then can use the score to predict depression in this new test cohort. And then there are prediction metrics such as R-square or AUC, which can tell you how good your prediction is. So it turns out that this help-seeking measure, this shallow phenotype of depression, GP psi, does great at predicting depression in other cohorts. And it's not just because with larger sample sizes, we're including more SNPs above of whatever p-value threshold. Because here I varied the p-value threshold, and basically in this panel here, I've included everything in the genome. The p-value threshold is 1. So having included everything, why is it that the shallow phenotype still does better than the deep ones if their effects are so non-specific to depression? If they're supposed to harbor lots of noise, how is it doing better than the deep version? No. So this must mean that better prediction does not equal to better specificity. And we need some way to disentangle the two concepts. So how do we tease those two apart? And in order to think about this effectively, we really need to start thinking about another thing, which is called pleiotropy. So pleiotropy occurs when one gene or one genetic variant influences many traits. And for a disease that is as polygenic as depression, you'd imagine that a lot of the genes that are involved in depression are influencing multiple traits. Yeah? So the PRS that you derive, the polygenic risk score that you derive from a genetic study of depression, is going to be predictive of more than one trait. It's going to be predictive of more than one thing. It's going to be predicting other phenotypes as well as depression. So hopefully, if your phenotype on this end is good, if your GWAS is really specific for depression, when it comes to predicting other things, it will predict depression much better than it predicts everything else. How well it predicts these other things should be dependent on how much genetic sharing they have with depression. Yeah? So that determines the difference between these two R-square values. Now, if your GWAS on this side is not so good, what would happen is it would improve prediction here by some degree, but it would disproportionately improve prediction in these other things because of the whatever genetic effects from these other traits is spilling over in this GWAS because it's not specific. So using this kind of relationship between PRS, like the prediction R-squares for all of these different traits, we can derive a metric called PRS pleiotropy, which is these R-squares divided by the R-square for the phenotype that you actually care about, which is depression. So if you did this across the phenome, if you did this across a lot of traits, hundreds of traits, you get 100 of these values, each one indicating how well this GWAS predicts the trait you don't care about versus the trait that you actually care about. So in this cartoon, I show some data points that makes up PRS pleiotropy distribution. So let's say we did GWAS on lifetime MDD, the deep version of depression in UK Biobank, 67,000 people. And the phenotype we are predicting is also lifetime MDD is itself. We do this on a tenfold cross-validation approach. So then the PRS R-square the PRS pleiotropy will be 100% because it's predicting itself. The top number and the bottom number is the same thing. So then it'll be 100%. And then let's make it predict depression symptoms, which I call DEP-all, and at some lower value. And then if I use it to predict, have you gone to the doctor for depression, is some other lower value, and neuroticism is some other lower value, etc., etc. So I can rank all of these things by their PRS pleiotropy and get a curve, basically. I can do this across loads and loads of traits in the data set that I have. So then let me show you real data. This is real data performed on UK Biobank across around 200 traits. I've cut it off at 60 because, going that way is just zeros. So the black line is for lifetime MDD, and you can see it's taken on the shape that we've seen just now in the previous plot. Lifetime MDD is here at 100. I didn't plot it. But then you see this curve coming down, and that's what we call the PRS pleiotropy distribution curve. So then, if you then take GP psi, which is the shallow phenotype that we were looking at just now, that's the gray line. And what you notice is that the gray line is above the black line. What does that mean? That means that for all of these phenotypes that is predicted here, it's the ratio of R-squared between those phenotypes and depression is way higher. That means they predict these other phenotypes much better than lifetime MDD would. So if, let's say, this phenotype is a good phenotype, you want it to fall on the black line rather than be on top of the black line, right? So this is basically an indication that although these PRSs are great at predicting depression, it's also much greater at predicting so many other things, and that's not specific to depression. Yeah. So then we can make this even clearer by just dividing this by this line, and you get now this black line becomes horizontal, and you can really clearly see how different they are. I just want to say that the pink line is the GWAS done by 23andMe, and you can see how similar they are to the gray one, because the phenotypes are collected in a very similar approach. So does that mean the vast numbers of data collected by shallow phenotyping is useless? Should we ditch all of that then, and then go back to deep phenotyping and go back to sample sizes of a few tens of thousands maximum? And of course, that's not what we're saying, and because that would be a big shame. We have so much data collected from self-reports, from electronic health records, etc. What we need to do is to find a way to use them that doesn't compromise specificity. Is there a way that we could use them, leverage them, that that preserves specificity to depression. And that's something my collaborators and I have been working on for the past 2 years. So we do this with phenotype imputation. Basically, what happens when you get a big biobank data set or a big electronic health record data set is that you have this measure of, what we call deep phenotype for depression, the good one. But there's a lot of missing entries in this data set. And then you get all of these shallow measures that have fewer missing entries. And then you get other mental health diseases, bipolar, schizophrenia, anxiety, etc. And then a bunch of things on lifestyle, socioeconomic status, stressors, etc., etc. And all of these are somewhat correlated with each other. There's some correlation structure between all of these different phenotypes. Yeah? So then we can leverage the correlation structure between the phenotypes and perform a singular value decomposition using the software. We're using the software called Self-Impute. We basically took off-the-shelf software. And then you get factors and weights that can be then linearly combined to fill in all these blanks using the covariance structure between all of these phenotypes. Oh, sorry. In addition to this SVD approach, that's pretty much the simplest thing you can do. You can also do deep learning. And that's what some of my collaborators at UCLA have developed. So basically, they've generated this huge deep neural network. It's autoencoder architecture. And basically, they've tested showing that their method, autocomplete, does better than self-impute in most cases that they've tested. So what we do is, the first thing you can ask is, okay, we've done our imputation. Does this actually improve GWAS power? Does filling in the blanks, firstly, give you better GWAS results than just deep phenotyping? And so we've done the GWAS on imputed phenotypes from both approaches. For reference, I just want to show you again that lifetime MDD, just remind you that if you did nothing with it, you get 1 GWAS hit. Yeah? So now I show the results from self-impute. So here are 270,000 people who originally had missing values for this measure. And now when we do a GWAS using purely values that have been filled in with imputation, you get 13 hits. And then if you combine the ones that are filled in with the ones that were originally collected, you then get 26 hits. Now, what happens if you use the deep learning method? Again, the ones that were only filled in, you get 18 hits. And if you combine the two, you get around 40 hits. So massive improvements in GWAS power. And then the question, of course, is any of this actually relevant to depression? So we've done a lot of analysis, and we verified that they are relevant to depression. We've done a lot of replication studies, etc. But the two things that we were talking about just now, do they improve prediction? And is the improvement in prediction specific to depression? So those are the two things that we're going to look at now. So the first is prediction. So again, here we have R squared on the y-axis. Ignore most of the phenotypes on the x-axis. I just took this from the paper that we've written up. But focus on this one. So lifetime MDD is here. It gives a prediction R squared of not even 0.01 for itself in UK Biobank. In red are all of the imputed ones. So firstly, you can see that the red bars are all much higher than this bar, this yellow bar. And then GP psi is here. This is the shallow phenotype version of depression. And you can see that most of these red bars are also higher than GP psi. So then what it's saying is that imputation gives us a phenotype that does better in GWAS and better at prediction. Now let's look at specificity. So again, the gray line here is GP psi, the shallow version that we saw just now. And what we talked about just now was the higher this line is, the less specific it is. So it seems that whatever deep learning is doing is not doing that great. Somehow it's getting less specific than the shallow phenotyping. But the SVD version is doing great. It's generating more power and better predictions than the shallow phenotyping version of depression, while preserving specificity better than the shallow phenotyping version. So then the lesson here is, we probably can improve upon all of these imputation methods. These are the first things we tried. We didn't really try very hard, to be honest. Phenotype imputation can improve power without compromising specificity. And we can optimize this for individual diseases, etc., in order to use the shallow phenotyping measures that we can so easily obtain from self-reports and electronic health records. In fact, there is a chance that imputation can even help us identify which of the GWAS variants from the shallow phenotyping definitions of depression are specific to depression. And why do I say that? So remember I said in performing a singular value decomposition, you get factors from the genetic covariance of all the phenotypes. So each of these factors is actually capturing the genetic covariance between some of these phenotypes. And in turn, is also capturing genetic covariance between some of these phenotypes. So in other words, each of these factors are actually indexing a pleiotropy axis between depression and all of these other phenotypes that went into the imputation. We can therefore characterize all of these factors, find out what their genetics are, and regress out each factor from this shallow phenotyping version of depression and see if that will help remove some of the shared effects between depression and all of these other phenotypes that we were talking about just now. So here I show you the percentage variance explained by the top factors. You can see that the top 10 factors basically explain almost all of the variance in this phenotype table. Here I show you what the top 5 factors from our phenotype imputation run captured. So factor 1 captures mostly neuroticism, factor 2, age-related things, factor 3, socioeconomic status things, education attainment, etc. Factor 4, again, socioeconomic status things, and then factor 5, mostly gender and sex. So what we did was then we wanted to regress some of these factors out of this GWAS. This is the shallow phenotyping GWAS that we said was not specific to depression. Just to remind everyone that GP-Psi, if you did nothing to it, it gave 18 GWAS hits for this in GWAS. So then regressing out factor 1, now we're left with 1 hit. We're like 17 of those are gone. And this 1 hit is on the gene NEGR1. And NEGR1 has been previously found to be GWAS significant for depression in pretty much all of the meta-analysis that has been done so far. And interestingly, it also has robust genetic associations with things like BMI, obesity, etc., a lot of metabolic traits. So what did we do to get this hit out of the 18 hits from GP-Psi? We regressed out factor 1. Factor 1 captures mostly neuroticism. So what this is saying is that whatever genetic effect is captured by this NEGR1 signal for depression may be independent of those that are tagged by the neuroticism axis. So we don't know the significance of this now. This is really the beginning of this kind of analysis. But I think this is one of those interesting downstream analyses that we could follow up on. Does this actually mean that there are 2 subtypes of depression, one due to metabolic issues and one more related to neuroticism, etc. So as I said, this approach is really, really new. Needs to be much more refined and tested to make sure it's robust. But it is the first principled way to identify specific associations to depression from the ones that are non-specific. And further efforts along these lines will be very, very helpful for us to prioritize genetic findings for downstream analysis. And I've got around 15 to 20 minutes left. That's not all. I will now, we've talked about GWAS of depression, but that really, I'm afraid, is really the tip of the iceberg in the complexity of this issue. So in the next 15 to 20 minutes, I will give a very brief overview of some of the other problems that we've started to look at with the currently available data, and that really needs a lot of effort solving. And the overarching theme of all of these problems that I'm about to talk about is that depression is a heterogeneous disease that is very likely the common outcome of many different biological pathways. So to tackle this massive problem of heterogeneity, we have to start somewhere. And the natural thing to do is to start stratifying patients of MDD based on some characteristic that may be indicative of them being one subtype while others are another subtype. So one of the first things that one might do is to stratify people by sex, by age of onset of disease, by their genetic ancestry, by their environments, etc., etc. So this is one of those things that people could do. The other thing that is really reasonable to do is to stratify people by whether they had severe stressful life events in their lives, because stressful life events is a strong predisposer to depression, either immediately or later in life if the stressors were in childhood. So let's look at the first one. One study that has actually examined sex population differences and recurrence in depression genetics is this study published in 2016-17, where they compared the Chinese cohort that I was talking about earlier in this presentation, Converge, against European-based cohorts from the Psychiatric Genomics Consortium. And what they found is that, firstly, genetic effects from Chinese cohort doesn't predict doesn't predict depression very well in European cohorts, and vice versa. They just don't do well at all. But because Converge is done only in females and only in recurrent depression, it predicts females in European cohorts better than it does everyone. Yeah. So what that says is that the effects from Converge are more specific to females in the Europeans, and it also predicts recurrent MDD in the Europeans much better than it predicts the whole group. So then this is saying that sex and recurrence are good indicators of some level of heterogeneity in depression. And the same is true vice versa. If you were using European effect sizes to predict a depression in Converge, then if your GWAS was performed on recurrent MDD, then it predicts Converge better. Let me see, where is the cursor? Okay, there we go. So more recently, a lot more trans-ancestry studies have been performed, and this is from the Million Veterans Project, where they looked at effect sizes obtained for genetic studies on depression in European cohorts versus those in African American cohorts. And what is saying is that just above half of the GWAS hits from European ancestry have the same directions of effect in African American cohorts. In other words, it's slightly more than chance, and that's really not that much correspondence between what we found in the European population versus what we find in other populations. And this can easily be interpreted as evidence for a little sharing between genetic basis of depression in both populations. However, what is true is that there's a lot of factors that goes into this. Perhaps the depression phenotype was collected very differently in the two cohorts. Maybe there are cultural differences with respect to self-reports of depression, etc. And there's a lot of different reasons why this could have resulted in such a difference. And this is supported by another recent study which looked into genetic correlations of depression between in East Asian ancestry and European ancestry. So they looked at genetic sharing between depression and other disorders. And you can see that although the sharing is quite similar for psychiatric disorders, at least they're all in the same direction, the directionality is quite different for some of the metabolic disorders. And this is indicative of, again, like potential cultural, social differences between the two populations that has created ascertainment differences in the data collection that has resulted in genetic differences. And much more studies need to go into this kind of analysis to firstly understand what depression is in each of the populations and what's the difference between them, so that we can make genetic research, and the findings from genetic research much more equitable across all populations. At the moment, there is still a very big Eurocentric bias in genetic studies, but it's becoming better, and hopefully with more efforts on this front, we're able to understand depression much better in all the populations. So overall, these studies across ancestries have become more prevalent. There is still an overwhelming need to have more diverse ancestries going forward, and we really should be understanding disease architecture in each population so that the results from genetic studies can also benefit them. So we've gone on to stress now, sorry. And so this is the other context that, quite often people stratify depression patients on. So we've shown in a previous study that cases of depression report a much larger number of stressful life events than controls. And we've also previously shown that if we remove the individuals with previous stressful exposures from GWAS on depression, then we can identify new hits that were previously masked when you perform a GWAS on everybody. So this shows that genetics of depression with and without stressful life events may be slightly different. And This is corroborated in many studies. However, the strange thing is, most studies, while they say the same thing in the sense that stressful life, if you stratify by stressful life events, those with and without have different genetics, the directions of effects is inconsistent. So this study is saying that the genetic liability of people with stressful life events is higher than those without. While in my study, I found that the genetic liability to depression in people with stressful life events is higher. So they're completely opposite in terms of directions of effect, but they agree that stress matters. So why do we have inconsistent effects when in this kind of analysis? And again, I think it comes back to difference in phenotyping. So maybe the stresses are different, maybe the depression phenotypes are different, maybe the ascertainment criteria for people coming into the study are different, generating all sorts of bias. So going forward, if we want to take environmental context into consideration when we perform genetic studies, we need to make some effort into establishing equivalence between exposures from different cohorts and sort out ascertainment biases inherent to each cohort so that we can find things that are comparable between cohorts and we can find genetic effects that are consistent and conclusive across them. So finally, we need to talk about heterogeneity in the manifestation of the disease across different individuals who have it. So firstly, depression patients exhibit many different symptom profiles. And according to the DSM criteria for diagnosing clinical depression, you could fulfill the criteria, well, two people could fulfill the criteria and share no symptoms at all. So there are numerous ways to meet this clinical criteria for depression. And there's a lot of subtypes that have been proposed through clinical experience, and they are now currently specifiers in the DSM for depression. Also, in addition to different symptom profiles, there's also different episodicity, onset, duration, occurrence, etc. In how people get depression. So, again, some recent work. This is a paper published last year, and it shows that depression with different symptom profiles, as indicated by some of the established clinical subtypes, have different heritabilities. So you don't really need to look at the words, but basically these bars are of different heights. And they also have different genetic correlations with other clinical characteristics of depression, such as recurrence, age of onset, anxiety, etc. One of those clinical subtypes is atypical depression, and that's characterized by increase in appetite and hypersomnia instead of insomnia, so you sleep more instead of sleep less. And this study from 2015 showed that atypical and typical depression have different genetic bases because the former has greater genetic sharing with metabolic traits such as BMI, triglycerides, C-reactive proteins, etc., while typical subtype of depression more often have genetic overlap with psychiatric traits such as schizophrenia, bipolar disorder. Another example of this, there's many studies on childhood onset depression, and this one in particular demonstrated that those with early onset depression have greater genetic sharing with psychiatric disorders, commonly thought to have neurodevelopmental origins such as schizophrenia, ADHD. But the thing is, these existing subtypes, they are great starting points for investigations into heterogeneity of depression. But relying on them really precludes any new insights on the structure of depression as a disease. You can study them, but you can stop there because there's no new subtyping coming out from that kind of analysis. So, as a response to that, people have started looking into symptom level investigations. They're looking into genetic contributions to individual symptoms, how they may vary across contexts, as well as their genetic overlap with other conditions. For example, factor analysis have also been done on the genetic covariance between the depression symptoms, and the clusterings has been done in attempts to recluster symptoms for MDD. Also, this reclustering using factor analysis has also been performed between depression symptoms, anxiety symptoms, items of neuroticism, to attempt to maybe redraw diagnostic boundaries, if that makes sense. In this case, it doesn't, because depression all clustered together. So in my group, we are also interested in looking at symptom level genetics. But one problem with phenotyping immediately jumped out at us. Which set of symptoms should we be choosing to look at? There's two sets, just in UK Biobank. There's many more, I guess. One is the CD short form, which assesses worst episode depression symptoms. It follows a skip structure. So basically, you'd answer the first two questions. Have you ever had a prolonged feeling of sadness or depression? Have you had prolonged loss of interest in normal activities? And only when you say yes to one of those two, you then answer all of these other questions on the other symptoms of depression. And basically, they're asking what happens during a worst episode of depression. That's CD short form. There's another version of this which says, over the past 2 weeks, how often have you been bothered by any of these symptoms? And there's no skip structure. Everybody answers everything. So, in other words, what's happening is that CD is enriching for a more clinical population because of this skip structure. And PHQ-9 is basically a general population survey on how you currently feel. So all of the symptom level investigations that I've shown you on previous slides are using PHQ-9. Maybe one of the reasons has a larger sample size because everyone answered it. This one has much smaller sample sizes. So if we wanted to start looking into depression heterogeneity, which one should we be using? And so we looked into it. My student Lian Yun looked into it. Firstly, she looked into genetic correlation between some of these symptoms with some corresponding traits. So for example, sad mood, maybe the best corresponding trait is subjective well-being, sleep changes, insomnia, worthlessness of feeling like there's meaning in life, and suicide ideation versus life not worth living. And you see that for all of these genetic sharing analysis, the traits always have greater genetic sharing with the current symptoms than with the worst episode symptoms. Okay? There's another analysis that we did. She then asked if these traits could have caused the symptoms. They could have caused the current symptoms of depression or the worst episode of symptoms. And she used this method called Mendelian randomization, which I'm not going to go into today. But it basically tests for whether the genetic effects on all of these traits are having the same effects on these other symptoms. So what she found was that almost invariably these traits significantly cause the PHQ-9 symptoms. Sometimes they also cause the worst episode symptoms as assayed by CD, but the effect sizes on PHQ-9 is always larger than those on worst episode symptoms. So Lian Yun performed a bunch of other analyses, but our general conclusion is that current symptoms as assessed in PHQ-9 are more indexing general dysphoria. That seems to be more trait-like than a state-like issue during a depression episode. And this could be corroborated by the fact that if you look at genetic sharing between all of the PHQ-9 symptoms, they are really high. They are around 0.8, 0.9, etc. This is genetic sharing between all of these PHQ-9 symptoms among themselves. And the genetic sharing across worst episode symptoms is much lower. So that's basically indicating to us that all these are very similar things, whereas all of these are quite different things from each other. Yeah? So then if you did symptom level genetic clustering using these two sets of symptoms, you find very different structures. So here I've done the factor analysis on worst episode symptoms as assayed by the CD short form. And this is the current symptoms as assayed by PHQ-9. And what you can see that this structure is reminiscent of quite a lot of the factor analysis done in twins and done in other phenotypic studies before. But this one, firstly, there are two factors, but the two factors are correlated at 0.9, so they're really, really similar. There's a lot less structure in PHQ-9 as compared to the worst episode symptoms. So what we learned from this is that going forward, if we wanted to look into heterogeneity during a depression episode or during one's, actual depression as a disease, we would be using worst episodes rather than current symptoms of depression, because current symptoms seems to be indexing something that is more trait-like than state-dependent. So we have two projects along these lines that are ongoing. I promise these are the last two slides. So one of them, again by Lian Yun, asks if the genetic effects between two subgroups of cases of depression, so these are subgroup 1, subgroup 2, both have depression. And for example, this one has symptom sadness and this one doesn't. Okay? So it's asking if genetic effects between the two subgroups of depression are correlated with the genetic effects between cases and controls of depression. So if they are, it means that the biological pathways that are distinguishing subgroups of people with depression are the same as those that are leading to disease, meaning these subgroups are etiological subgroups, and we can use those to identify biological pathways leading to disease. There's a second project that asks if the pathways leading to different symptoms of depression are acting additively or in interaction with each other to cause MDD, and whether we can learn anything about depression through that. Because at the moment, the symptom criteria for depression is looking as if it's all additive, but maybe there are some interactions going on too. We're getting quite close to finishing some of these projects, so hopefully if I ever see you again in the future, I'll be able to tell you about this. So stay tuned for the answer to these questions. So finally, what we talked about today, phenotyping matters when it comes to genetic studies. For depression, it matters quite a lot. Imputation can leverage both shallow and deep phenotyping to get the best of both worlds, and this is something that we probably should be doing a lot in the future as different types of data come in from electronic health records, medical registries, prescription registries, etc. There are methods for identifying specific from non-specific genetic effects, and these could be better developed in the future to really robustly identify specific genetic factors for depression. Depression heterogeneity is the next big challenge. There's a greater need for diversity in GWAS and a need to characterize environments properly and need to validate both clinical subtypes and perform symptom level analysis to more agnostically find out if there are newer subtypes potentially. So thank you very much for your attention. I've been going on for really long. It's 1 hour exactly. I'd like to thank my group. Like, this is my little research group in Munich. If anyone wants to join, I'm actually hiring postdocs. If anyone's interested, please let me know. These are my collaborators who I've done all of this work with. So thank you for listening, and I'm very happy to take any questions. Thank you.
Many thanks indeed for introducing us to all those complexities that we didn't realize went into research on the genetics of depression. We normally now have a little break while those who have to get away can slip out, and then we have some questions. So.
Should we stop this? Should we click stop?
If we get some interesting questions,
maybe we can slip out and not be on camera for the break.
Lots of things. So, questions for Narchai? Yes.
Fantastic talk, thank you very much.
Thank you.
I really like the sort of analysis you were showing towards the end, sort of thinking about clusters of symptoms in different depressed patients. And you had some differences there depending on the measure you use, whether you were sort of looking at the worst episode they'd ever have or sort of current episode. And you sort of had this interpretation that could be state versus trait, is that right?
Yes, that's right.
Could another explanation be sort of what, in one case you're relying more on memory? So if you're asking about the worst ever episode, that could have been many years ago, where that doesn't exist with the, if you're talking about symptoms you currently have. Have you thought about that?
Yeah, I think so I don't completely understand it myself, to be honest, but and I think the extent to which we are thinking about it now is probably not the full extent of it. But how we're thinking about it is this. If you're asking someone right now whether they're feeling one way, the people who are likely to say yes to that are probably likely to feel that way a lot of the times because you're taking one cut in their lives. The people who are more likely to feel that more often is likely to be the ones who say yes then. So then that's why we are calling it trait-like. It's not the best term ever, but but that sort of captures some of what we're trying to say. It's just that they're more likely like that more of the times, which is more trait-like, yeah.
I see. Because it seemed like the effect sizes were weaker, right, for the worst, when you were looking at the worst episode measure, but a lot of them were still seem in the same direction. So again, that might be sort of just a noise thing. And
Exactly.
Rely on memory.
That's true. That's true. Yes.
Deborah.
I don't understand almost anything about genetics, but you had a very, very clear talk.
Thank you.
I'm going to try to ask you a layperson's question.
Sure.
But apologies if it's based on a complete misunderstanding of what you said.
No worries.
It seems that the majority of evidence that you presented suggests that depression is a nonsense label that has no value, and that we need to start afresh and think about it, that coming from genetic research and all of this heterogeneity, if there was really something in it, all these clever people would have picked something out of it. So is that a good take-home message?
You're really polite with that question, I think. I think what I often struggle with this when I'm doing the research as well. Given that the label has been there for such a long time, that depression has been defined like so long ago, and all these diagnostic criteria have been made so long ago. It hasn't really changed almost at all since the 60s or something. Who am I to, as a person who's doing research now, like how can I question all of it? Can I redo all of it and find new things? And that's something that I feel like shouldn't stop anyone from looking. I mean, there should be some level of healthy skepticism that allow yourself to be doing certain analysis that may show you new things that you didn't know about before. But I wouldn't go as far as to say that the label's completely meaningless, because before we have anything new that is better, that's still the best standard that we can use right now to deal with it. So we'll work with it until we know that there's something better out there that we can define that works better than the label that is currently available, I think. Maybe that's the right sort of, or maybe that's a balance that we could aim to strike, and that would be helpful.
Do you have a lead towards something that you think is looking like a potential candidate? Because if it's not the same in one country than another country, in one race than another, people don't share symptoms, there are lots of subtypes. People looked with hundreds of thousands of people and haven't found any genetic. It's not actually there.
Well, that would be like. So the GWAS on hundreds of thousands of people, or millions of people, they found a bunch of things. The problem, as I've said just now, like and you've taken away so well, is that we don't know which is which. So some of these hits may be for one subtype, some of these hits may be for another subtype, some of this may not be depression hits at all, they may be for other things. So we don't know which is which. And with more data coming in on more clinical characteristics of depression, treatment responses, comorbidity, environmental data, etc. So all of these are not previously available to us to make any distinctions between different groups of people who may be having depression. So I feel like now why I say heterogeneity is the next big thing is really because we now have much more data than before to even look into it. Previously, when we were struggling with sample sizes to find any genetics at all for depression, like what people were doing is lumping everybody together so that you get like sample sizes in order to get some level of genetic power, like statistical power to find anything at all. But now with these large data sets, if sample sizes become less of a concern, if we have much more data from everywhere, then firstly we could stratify people much better into like subtypes and look into that. And secondly, even the massive differences between different populations may be really interesting, because that could tell us what is happening here, what is happening there. Are there different ascertainment biases? Are there different cultural issues? Are there different health system inbuilt issues that are causing these two populations to have such different diagnostics, etc.? For example, if one hypothetical health system works off this diagnostic standard, while another hypothetical health system works off another, then they would give different phenotypes that would lead to quite a lot of differences in genetic findings. And that would be interesting to know to begin with in itself, and working based on that with all of the population-specific environmental factors, etc., could be really a good thing, because then we're not using what we find in one population to define this phenotype in other populations and claim that it could help them translationally. I think that would be really what we are hoping to do next. So I wouldn't write it off completely right now. Like it's still useful to have those labels for as long as we can work on them. If we ever find anything that is better, I think maybe then, well, firstly we have to prove it really robustly, and then, all of that. Then maybe we revise it. But I think the possibility to revise is something we could keep in mind. Like we shouldn't stop ourselves from doing this kind of analysis.
Thanks for a really, really nice talk.
Thank you.
To think about. I was curious about your imputation and the sort of factors that led into it. So there was neuroticism.
Yeah.
And then subsequently a lot of the factor were SES-related variables. Now, how do you sort of make sense of that? Is that almost a function of the fact that UK Power Bank has a whole range of SES-related variables, so hence they will sort of feed into that? Or are we sort of capturing something in the environment which sort of then sort of correlates with our biology and genetics quite a bit? And in which case, what are your thoughts on how do we sort of integrate innate genetics with the complex dynamic environment to then sort of lead to sort of subtype and heterogeneity discovery in depression?
I think that's a great question. I think your interpretation is exactly right, that some of these factors are definitely capturing environmental things that are correlated with genetics. That so ideally we don't include them in genetic studies, then ideally we regress them out from GWAS, etc. But the way to do that is really complex, because as you've seen, what I showed you just now isn't going to be robust in every situation. You shouldn't be regressing out heritable things out of GWASs. That would just give you collider bias immediately. So then there's like careful ways to do it that we haven't developed. We should be developing those. We should be thinking about how to do it properly. So there's a lot of new things that needs to be done for that to work properly as intended. And that, like you said, is well, in terms, when we were doing the imputation in UK Biobank, one of the things that we, by design, we weren't very careful in choosing the phenotypes that went into it. It's literally whatever I can put my hands on, and that's easy to pull out of the touchscreen questionnaire. I didn't select based on genetic correlation. I didn't do anything. It's like whatever I could easily figure out, like put together. And that's almost by design, because you want it to be a more generalizable process where you don't need to be an expert on the disease or an expert on the whole environmental architecture that leads to the disease before you can start doing such an imputation. It's almost meant to say that if you don't know the disease at all, you can still do this and it will still give you something. But that's not really utilizing the biobank to its fullest, because there are all these other phenotypes that we didn't include. We haven't included omics, we haven't included imaging, we haven't included comorbidities, we haven't included ICD-10 codes of anything. So one of the things that we were thinking about doing, like in the future, or other people could also do in the future, is would be very interesting to see which phenotypes going into imputation would give us the best answers, would give us the best imputation that is the most specific to depression. And from doing that, we can even tell, like, what are those phenotypes' relationship with depression itself, and that tells us quite a lot of things about that too. And we're specifically particularly interested in those phenotypes that could help us improve portability across ancestries. Because if you think about it, like one of these hypotheses that I have is just genetic effects could be differing between populations purely because of allele frequency differences, because of drift. One allele is present at such high frequency in one population, even if it has a huge effect on the disease, if it's present at 0.2% in the other population, it will just never show up in any of the association studies. But but the biological, so individual genetic variants could be very different, but biological pathways should be much better preserved. So whatever is on those biological pathways, be it environmental factors that affect it, etc., they should be much better preserved across. The whole structure of whatever's leading to disease should be better preserved across different populations. So if we were able to use, if some phenotypes going into imputation gives us much better portability across ancestries, well, that would be those invariant risk factors for depression that you really would be caring about. So why wouldn't it be nice to identify those using this approach? Maybe it's a pretty roundabout approach. Maybe you don't need imputation to do this. But that's one thought that we've been having, and in terms of how to think about these factors and these phenotypes going into imputation.
Thanks for a fantastic talk.
Thank you.
I was wondering, with the societal changes in terms of how we relate to stigma versus acceptance and so on, how much effect do you think that might have on historical databases and the data that you use?
Oh, that's a wonderful question, I think. That is really interesting, and we are only now beginning to have any data to look into that, because it's only now possible to look at it because we are starting to get access to medical registries and electronic health records, which have the diagnosis from many, many years ago. Prior to this, it's almost all cohorts which are collected at a certain time, about what's happening at that time. So now it will be really interesting to see if, for example, validity of diagnosis, for example, genetic contribution to diagnosis were different for diagnosis made much earlier on than now. This could be different between different disorders. Some disorders may have been established for longer, maybe the diagnostics haven't changed for a lot across the years, then maybe this doesn't matter for those diagnoses. But maybe for some other psychiatric disorders, maybe the diagnostic, the label didn't even exist years and years ago. So then, surely there were people suffering at the time. Maybe they were mislabeled as something else. So this is something that we're interested in looking at going forward, using data that dates back quite a while, rather than just whatever is available now. So I think it's a really great question. I think it's really interesting to be looking at, and especially when you think about things like comorbidity. Like if a person has more than one diagnosis across time, are some of those not right? Especially if they were made really early on, before that particular label is available to them. So that's really interesting.
Yeah, thanks so much. I learned a lot from all your explanations.
Thank you.
I was curious about how, in the first part, you were using specificity as the sort of criteria for whether we're getting closer to the genes that might lead to the biological pathways that we want. And I guess I want to ask about why is that a good criterion, given that, you just said about how there's comorbidities, and that's particularly high for depression, as I understand. And so why might it not be the case that the biological pathways that we're looking for depression are also implicated in a whole bunch of other things like neuroticism?
That's a great question also. I think that's a great question. I think what is the criteria for specificity is really on the sort of study design level. We're not saying that genetic findings or genetic variants or genes or even pathways that are not specific to depression are not relevant to depression. They could be, exactly. Depression does have a lot of genetic sharing of other diseases. That could be biologically true, and it would be really interesting to look at. But in terms of if you were to design a study and if you were to sort of decide which phenotype you want to be looking at, having a more specific phenotype for depression is going to help you. So that's the pleiotropy thing that I was talking about just now, isn't it? Like depression has natural pleiotropy. Like the genetic factors that are leading to depression has pleiotropy with a bunch of other things. But how do you know when this phenotype that you're looking at is having excess pleiotropy as with all of these other things because of misdiagnosis, because of noise, because of poor phenotyping, etc. So that's really difficult because am I going to draw this line here? Like, this level of pleiotropy is fine. A bit above it is not fine. that's obviously like quite a difficult line to draw. So and also it could, it's really difficult to define, I think. So the reason why we're looking into specificity in all of those earlier slides on GWAS, etc., is so that we don't have to feel like this line is floating above what could be true. It's sort of almost like how confident you would be in all of the genetic findings that you get. Are they going to be mostly due to misdiagnosis, or they're going to be mostly due to depression? If there's like, let's think of hypothetically, there's like gold standard for depression. There currently isn't. You can't. There's no way to do this. Well, maybe if a clinician's structured interviews, administered, etc. That's the best we have right now. What did I want to say? So, I think if you knew that this phenotype is poorly defined, it contains a lot of misdiagnosis from other places, its heritability is way lower than what you get from a clinician's diagnosis, then you'd be so much less confident in all of the genetic findings that you get. And then if you work with that in terms of comorbidity, in terms of heterogeneity, etc., you're likely, the issue with that is you're likely to find things that you not normally find for depression. That doesn't help depression. So you can't follow it up with anything. You can't confidently say that, okay, well, here's a drug target. Here's something that I want to put resources into cellular studies, mouse studies, clinical trials, etc. Can we make a drug out of this, or can we make a biomarker out of this? It's very hard to have any sort of confidence going into those follow-ups. Whereas if you're a bit more confident about the phenotype, you know that it's a bit more specific to depression. Whenever we talk about specificity, we always reference it to some level of standard. And the version that I had in my slides was the deepest possible in UK Biobank. That could also be a clinician interview cohort. That is of enough sample sizes. But so you're always referencing it to something which you are calling the gold standard. Now, the gold standard may not be right, and then that's something that, other kind of analysis could improve. But at least there's some standard, at least, we're all talking about the same thing. And I think that's helpful. Yeah.
Yeah.
Just wanted to say some amazing. It was absolutely amazing. Really enjoyed the talk. And I think my question was more so, I think diagnosis for neuropsychiatric disorder is such a complex issue, and then there's treatment.
Yes.
And I was wondering, how does this finding help us develop new therapeutic strategies? Because I think currently the practice is still sort of treat symptoms rather than, or theories which were we know from decades ago, right, that there hasn't been any new improvements per se. There's been drug repurposing to some extent to sort of figure out what can new ways to sort of approach this. I wonder if sort of what the lovely lady touched on over there, sort of removing these labels, so to say, and then more so focus on pathways or biological significances we've identified to sort of drive this therapeutic side of things. And I guess I wanted your opinion on that.
So I've never, I've never worked anywhere close to seeing any of the hits to the therapeutic side. That has just not been part of my job at all. So I have very limited opinions on this, I think. But I think one thing that is important for drugs is that you cannot have massive side effects, which is a big problem now for psychiatric drugs, antidepressants, etc. And I wonder if it's, at the moment, most antidepressants, we don't actually know how they work. They work in some people, but not others. We don't know why. Going forward, if you were to develop drugs, this comes back to the specificity issue. If this, for example, gene is targetable by something, by a chemical, let's say, and we've shown that it's associated with depression over and over in different cohorts, etc., etc. One thing is, if it's also associated with a bunch of other things, like metabolic disorders, immune disorders, etc., then it's so much more likely that any drug that is targeting it is going to have other effects somewhere else, and that's going to make it very difficult to, it will make it difficult for someone to keep taking it and not have other issues, for example. I mean, this is like such a sort of cartoon example. I mean, I don't know if the actual drug targets that we might end up identifying are of this sort, but in general, the idea is that in identifying potential drug targets, they should, as much as possible, be specific to the disease that you're looking at, and you should probably know what it does in cells, etc. So that's, I guess, another reason to think about specificity. What do you actually want to do with those genetic findings later on? Do you want to make drug targets? Do you want to make diagnostics? So one thing that I didn't talk about today was in my first project on depression, we used sequencing data. We found this biomarker. Biomarker, like it's a molecular signature. Like we found that a mitochondrial DNA copy number is elevated in people with depression as compared to controls. Okay, so on the first look, it's like, okay, definitely artifact. Definitely something has gone wrong there, and could be because of whatever artifacts that happened during sequencing. Tried to make it go away, signal didn't go away. Replicates in 3 or 4 other cohorts. Done animal studies. If you stress the mice really hard, you can see mitochondrial levels just rising. Okay, do we have now a biomarker for depression? Can we use it now in clinics? Can we take somebody's blood and measure mitochondrial DNA, put it on some population distribution, see whether they're high or not? Can we use this as a metabolic indicator of someone having depression? And of course, no. Subsequently, after we published this, lots of people have found that mitochondrial levels increase because of whatever. And they're like, okay, well, then maybe it's not specific to depression. Maybe you can't use it for diagnostics. It's useful to know that it happens. It could lead you to investigate biological pathways that might be involved, either as a consequence of depression or as a cause of depression. We don't even know that. So it's interesting to know about this on the side of learning about biology of depression. But what it takes to get something into the clinic, I think there's a lot higher in terms of requirement. You need to be specific. You need to be, maybe only working in one tissue. Like this phenomenon may only happen in whatever cell it is that precipitates this this symptom or biological issue that led to depression. So I think there's a lot more that needs to be done before we are anywhere near there. Yeah.
Just better to the last question as we come to second.
That made me think of another question. Has anyone done GWAS and stuff on mitochondrial DNA?
Sorry, what's that?
Has anyone done these kind of studies but on mitochondrial?
I have done it, but I've done it on the Chinese population. I can't find a replication sample. Absolutely, it's sitting on my computer now. I've got hits, but I don't know. The mitochondrial genome is so different from the nuclear one because different populations have such different variants. Because of migration and stuff like that, it's only passed down maternally, no recombination. So the variants that are present in the cohort that I was looking at are just not present at all in Europeans. I've tried doing mitochondrial analysis using UK Biobank as well, but UK Biobank currently, it's now doable. If anyone wants to do it, you can do it. They've just released whole genome sequencing on, I think, a portion of UK Biobank. I don't know when the rest is coming. But with sequencing, you can call mitochondrial variants really confidently because usually, I don't know how much you guys know about sequencing, but when you sequence a genome, usually they talk about coverage as in how many times coverage. 4 times coverage means covering everywhere in the genome at least once. When you have whatever coverage in the nuclear genome, you usually cover the mitochondria about 100 times. So you've got loads and loads of reads on the mitochondria, and you can really, really confidently call variants. So if you get your hands on, whole genome sequencing data on the mitochondria, you can call variants really confidently, and you can do associations really confidently. The reason why I haven't done it in UK Biobank is firstly, at the moment, before sequencing data is released, all the genetic data is from genotyping chip. And the genotyping chip in UK Biobank is pretty good on the mitochondria. It has optimized for both common variants, rare variants, etc. It's made by experts who are working on mitochondrial genome, but it doesn't cover all of it. So I've done the association using what's available in UK Biobank, didn't find anything. Still holding out for the sequencing data. Maybe the sequencing data will tell me things that are not covered by the genotyping chip. So that's still something that I would be very interested in doing. Yeah. Sorry, that's a really long answer that you probably didn't ask for. But
It is 6, so we probably should stop
Yeah.
the official proceedings now. The Sangamo Club normally at this moment retire to all bar one in Regent Street, but Dr. Cha has to go to another appointment. So what I suggest is that if anyone still has burning questions, they catch her before she actually leaves, just inform me while we're packing up. But otherwise, we'll thank you very much indeed,
thank you.
for a lovely talk. Many thanks indeed.
Oh, thank you so much. Thank you. Really, really great questions from everyone.
Very useful questions. We could now